Good morning, everyone. It's nice to see a good turnout this morning again. Uh, I'm not a morning person myself, but hey, this is the class, this is the job, and this is what we all have to do to be responsible, right? So great to see you here. Thanks for joining. Um, so I got quite a few of questions about the participation exercise. So the questions were emailed to me, which is fine. But I want to remind to you that I have office hours on Monday and Thursday, and I spend my time answering your questions then. So I do think that answering questions face to face, talking it out, will be faster for both of us, and it will be more effective. Because if you hear the answer, I think it will be memorized more easily than if you just read about it. In addition, during office hours, I can share my iPad and draw a diagram for you, or share my screen and explain the image to you better rather than in text by email. So don't be shy to book a visit during office hours, and I'll share my screen and remind you how to do that. All right, so to book office hours, the office hours are conducted via Zoom, and this is the link to the Zoom room for the office hours. And please book the appointment here. So if you click on this link, it will lead you to my Calendly page, and it will show you when do I have available dates. So I have office hours tomorrow, and you can book the appointment at 12:30, 12:45, 1, 1:15, and so on. And I have a lot of appointments next Monday and next Thursday and the following one. So the dates highlighted in blue are the dates that I do have office hours. And once you click on the date itself, you can see the appointment times that are available. So don't be shy to book office hours. You may not think so now, but they are helpful. They are effective in making students learn, in making you retain the information better and even understand it better. Because if you didn't get a concept during the lecture, for whatever reason, you stepped out of the room, or you went over the recording and it's still not clear to you. So I can explain it to you again in a way that is more convenient for you. Or Ansley can explain it again. Ansley's office hours are also here. So to book Ansley's office hours, you can click on this link. Oh, and there's no openings at the moment. Ansley, I guess Ansley is all booked up, unless there is a glitch. All right. Um, okay, there's that. Because I got a lot of questions about the participation assignment too, I wanted to go over the correct answers and why are they correct. So the first question gave you a picture of the electron transport chain, and it said that as electrons move through the electrons transport chain, molecule X, this thing, is being oxidized or reduced, as molecule Y, this thing over here, is being oxidized or reduced. So you had to choose which variant was correct for molecule X and molecule Y. Now we have an electron donor molecule that donates electron to this protein, the first protein in the transport chain. What I think a lot of you missed, well, a lot of you did great on this question, but I got quite a bit of emails about this question, so I'll go over it again. As electron moves, pay attention to the direction of the arrow, okay? The electron moves from here, moves all along this yellow line, and eventually lands on the electron acceptor molecule. So the molecule that is being reduced accepts the electron. The electrons lands on the molecule that is being reduced, and the molecule that is oxidized donates an electron. So first the electron jumps here, then it jumps here. So if an electron jumps from the first protein to the second protein, the first protein becomes oxidized and the second protein becomes reduced. Now the electron jumps from the second protein to the third protein. As even though this protein was reduced just milliseconds ago, as the protein jumps from the as the electron jumps from the second protein to the third protein, the second protein now becomes oxidized, and the third protein becomes reduced. So this is our molecule X. Now the question was asking about this transition here, the transition from molecule X to molecule Y. So the transition from the third protein in the chain to the fourth protein in the chain. So even though molecule X was reduced just a minute ago, a millisecond ago, like when the electron jumped from the second protein to molecule X, as the electron jumps from molecule X to molecule Y, molecule X is oxidized and molecule Y is reduced. Any questions about that? All right, I guess not. Moving
Professor,
on. Yep, go ahead.
will the order always be reduced and then oxidized?
It depends on the movement of the electron. So pay attention to where electron is moving. So if I asked you if the, let's see, if the electron lands on the molecule is being reduced. If electron leaves the molecule is being oxidized. Does that answer your question? So always pay
Yes,
attention
thank
to
you.
the electron. The order may not, make sense. What makes sense is following the electron, like following where electron goes. Okay, I'm gonna mute you, Riley, if you don't mind. The second question. So I gave you the graph from the lecture, and I said the question was, according to the graph, at what temperature do you predict the bacterial enzyme becomes denatured or loses its properties because the temperature is too high? So I'm not asking about the human enzyme, I'm asking about bacterial enzyme. So we discussed the x-axis here is temperature, the y-axis here is the rate of reaction, which is mediated by the enzyme. The reaction starts here at this temperature, about 55 degrees Celsius. It achieves its optimal rate at about 73 degrees Celsius. So the enzyme is still present when at 73 degrees Celsius. But when temperature reaches 78 degrees Celsius, the reaction rate is low or non-existent. And this is because the enzyme, which mediates or catalyzes the reaction, to catalyze means to speed up a reaction. The reaction rate does not go anywhere because the enzyme is denatured, or it loses its properties because the temperature is too high. Any questions about this? Okay, the question, does enzyme start to become denatured starting at temperature after 73? No, there is a specific temperature that enzyme becomes denatured. For us, it's around 41 degrees Celsius. And for this bacterium from a hot spring, it's at 78. So whenever you see the rapid drop off, whenever you see the reaction dropping to zero or to very low here, because it's not numeric scale, this is when the enzyme is denatured. It's not a gradual denaturation. It's sharp drop off because denaturation is fairly quick process. All right. Any questions about this? All right. So in another question that I got in email was, well, not so much a question, but there was an email that kind of concerned me how some people view due dates for quizzes, and I just wanted to reiterate that quiz one is open now and it is due on January 29th, but you can take it anytime between the time the quiz is open and between and its due date. You don't have to take it on the due date. I know a lot of you do, but I don't think it's a good practice. Okay, you can at least start or take your first attempt way before the due date, so you can study and know what the questions are and know what you have to rewatch in the lectures and the supplementary videos, or schedule office hours with me and go over the concepts that are not clear or the questions that are maybe not clear. You can go over the questions with me during the office hours. That's not against the rule by any means. Um, so quiz two, again, this quiz one is due the day after tomorrow, this Friday. Quiz two opens immediately afterwards, so its open date is at midnight on January 30th, and it's due on Friday, February 5th, so in a week after it opens. Ansley, are you here today? No, no Ansley today. Well, I hope she's okay. So I'm going to go over chat when I can and answer the question when I have a break for questions. So, what is the material? What material is on quiz one? I was waiting until the Friday's lecture, so everyone was covered. Everything was covered. Don't do that. Don't do that because look at the quiz first. Okay. I never give out quizzes if with material that have not been covered at least last week. So when the quiz opens, the material for the quiz has already been covered. And if you're not sure what is covered for the quiz, just literally click on the quiz, and in the description of the quiz, you will get the materials that the quiz covers, and all of them are available on web courses. So the materials that you will typically need to study for the quiz is to review specific lectures, and I give you the information which specific lectures to review. And the second type of material is supplementary videos, and I give you the list of specific supplementary videos with links that you need to review to take the quiz. Okay, so don't wait until the due date because you're not sure that the material is covered. The material that is covered for the quiz is listed in the quiz description. Okay. Okay, I couldn't find lectures. You know where I can find them? Yes. So the lectures are located in the modules tab in the section for our class and web courses. So if you go to modules tab, you will see this elaborate schedule where it's separated into units. And we're right now in unit one. And unit one has a subheading called lectures. And if you click on the link, links posted under lectures, it will take you to the video of the lectures. And under each lecture, I also post the notes that are associated with the lectures, or PowerPoints. I see a question about the quiz. So the question is, in quiz one, there were a few questions that had the answer choices as were, you can select more than one answer, but was not specified if you were looking for more than one answer. If not specified, are we supposed to select more than one answer? You can select the answers that you consider to be correct, okay? If it allows you to select more than one answer, it means that you can select more than one answer. I mean, yeah, but yeah, you should be confident after watching the supplementary videos and after watching the lecture video, revising the lectures. It should become straightforward which answers to select if there are multiple choices available. All right, I am moving forward to the lecture. I'm going to pick it up on the energy of light from where we left off yesterday. So yesterday we roughly left around here. So I discussed the various ways if when we discussed various ways that substances or solutes can pass through the membrane in the cell. And I discussed that I mentioned that some of these mechanisms require energy and some of them don't, and some can even generate energy. So the pathway, the method that solutes pass through the membrane that does not require energy is just simple diffusion. And osmosis is a special case of the simple diffusion. So diffusion is applicable to any solute, and osmosis is applicable to water only. So if we have a vessel with a semi-permeable membrane in the middle, and consider the semi-permeable membrane an analogy for the cell membrane. We may have more molecules of water on one side of the membrane compared to the other side of the membrane. So if the membrane is selectively permeable or semi-permeable, like the membrane in the cell, it's if the concentration of water on both sides of the membranes will even out. So if there's more concentration of water on the left side of the membrane here, the water molecules will move through the membrane into the cell, or to the other side of the membrane, to the right here. So if sometimes the solute moves, like sometimes this solute molecule moves through the membrane, but then that would be considered diffusion. But if the water is moving through the membrane, it is considered osmosis. It is called osmosis. So osmosis is diffusion of water down the gradient of concentration. And again, the expression down the gradient of concentration means movement from area of high concentration to the area of low concentration. Ansley, did you join by any chance? Ansley is not joining. Okay, so what I'm gonna do, I'm gonna go through the lecture, and then I'm gonna try to leave the last 5 minutes for questions. Okay, and go over chat and answer the questions then. So moving on. So osmosis, because it's the diffusion of water through the membrane, osmosis determines water content in plant cells. Plants usually keep their solute concentrations lower inside than outside so that water enters the cells. However, hypotonic surroundings result in a loss of water, shrinking the vacuoles. So in this picture here, you see the cell, the cell wall and cell membrane. The little green things are chloroplasts, and the large blob in the middle is the vacuole. And remember, the vacuole is the organelle of the cell uh, that is analogous to lysosomes in the animal cell. So it digests stuff and it stores water. So if the surroundings of the plants are hypotonic, the plants will lose water. So hypotonic surroundings cause the cells to lose turgor pressure, causing the plant to wilt. So what it really means, turgor pressure is the pressure of water from the inside onto the cell walls. So if there is a lot of water in the cell is healthy, it pushes against the membrane, expanding the cell. So this pressure from the inside of the cell by vacuole, let's say, it is called turgor pressure. When turgor pressure is low, cell shrinks and the plant start looking sad. So why would the plants in the I'm sorry, I confused things. So in hypotonic surroundings, the water rushes into the cells And in hypertonic surroundings, the water rushes out of the cell. So in a hypertonic surroundings, it means the surroundings, the surrounding environment around the plant has less water than is contained inside the plant. So let's say if you have a potted plant and the air is dry and you haven't watered it in a while, then water will rush out from the cells, it will evaporate, and you will see that the plant cells will lose turgor pressure and become sad and wilted. However, in hypotonic surroundings, you may see that the plant is standing upright. If the air is very humid, or if you water the plant very well, it contains a lot of water in its between cells, and the water rushes into plant cells and causes the plant to look healthy and not wilted.
Can I ask a question really quick?
Yeah, go ahead.
On that last slide for the second paragraph, so should the hypotonic in that one be hypertonic? Is that like a typo? Because like I was just wondering, because the hypertonic you said is what loses water.
Yep.
Okay, I just want to make sure.
It is a mistake on mine. I will correct this before posting the slides.
No worries. Thank you so much.
All right. So this is another example how osmosis affects the cells, and this time it's an example not for plant cell but for a blood cell, red blood cell that course our bodies. So the red blood cells are called erythrocytes. And if you've ever had a blood test done, this is a very common thing to test for the quantity, the counts of erythrocytes per unit of volume. So within our bodies, water continuously moves into the cells and out of the cells, including the erythrocytes, the blood cells. If the solution surrounding the blood cells is isotonic, meaning there's equal amount of water around the cells as contained inside the cells, then the water will just stay in the cell and the water will be in equilibrium and nothing bad will happen to the cell. If the blood cell is located in hypotonic solution, in hypotonic solution, water will rush into the cells from the outside. So in hypotonic solution, water content around the cell is higher than the water content in the cell. So this causes the water to rush into the cell and change its shape, like make it misshapen to hold all the extra water. Now, if this surrounding environment contains less water than is contained in the cell, then water rushes out of the cell because water, according to osmosis, water moves down the gradient of the concentration. So in the case of the hypertonic solution, there is more water inside of the cell than outside of the cell. So water moves outside of the cell to balance out. So this kind of funky transformation is the reason why people are not just injected with water. Like if a person gets sick and dehydrated, let's say, and just drinking the water wouldn't cut it. You would need to place an IV into the vein. The IV itself does not contain water. If IV itself contained just water, we would lose a lot of red blood cells because there would be a lot of water in the surroundings compared to the cell interior, and the cells would swell and lose its properties, important properties of carrying oxygen. So we are not injected with water. We are injected with some specific saline solution to remain for the surroundings to remain isotonic, or for the environment inside our blood vessels to remain isotonic. All right, moving on from the diffusion type of transport from the molecules, which does not require energy, to the facilitated diffusion. That also does not require energy, and in some cases, it even produces energy, like here, like in this picture here. But facilitated diffusion requires proteins that are embedded in the membrane and that would allow the solutes to move from outside of the membrane to the inside of the cell. So the reason why, so in this picture we have H plus, which is a hydrogen ion. Hydrogen, the more hydrogen ion is in the environment, the more acid the environment becomes. So for instance, in our stomach contents is highly acidic, and it helps to digest food and kill all the bacteria in the food or microorganisms in the food to make it safe. So our stomach contents has very high concentration of this hydrogen ion, proton. So the hydrogen ion cannot just pass in between these phospholipid molecules in the membrane. And the reason for that is that in the phospholipid molecule, its head is hydrophilic and its tail is hydrophobic. So if it is the head is hydrophilic, the molecule, which is an ion in this case, it will get stuck to the head and will not move further down in between the phospholipid layers. So what it needs, what it needs is a facilitator. And this facilitator is going to be a protein, which is embedded in the membrane. The protein has a channel, a specific channel that is suited for the passage of a particular ion or a molecule. And this is the way that an ion or a molecule will get in. And in this case, you see that ADP, or adenosine diphosphate, is phosphorylated, or phosphate group is attached to it, and it forms ATP. And ATP, remember from the last lecture, is the energy currency of the cell, of our organisms. Any energy that is required for the work to be done needs ATP. Yeah. And the third type of transport of solutes into the cell and out of the cell requires energy. And this facilitated transport that requires energy is called active transport. Active transport always needs a protein embedded in the membrane, okay? And it always needs energy. So in this case, we see that we see the inside of the cell here, we see the membrane, and we see the outside of the cell with the high concentration of some solute or some substance. And we see that somehow the substance is not moving down the gradient of concentration, but moving in the opposite direction. So if it were just facilitated diffusion, the substance or solute would move from the area of high concentration to the area of low concentration, and maybe even produce some energy as a result. But sometimes we do not need a lot of particular ion or molecule inside the cell, otherwise it becomes harmful. So the cells work to expel that molecule or expel that ion from its insides. And to do that, it requires energy because it goes up the gradient. The molecule is moved up the gradient of concentration, or from the area of low concentration to the area of high concentration. This type of membrane transport where the protein is involved and it requires energy to expel an ion or a molecule is often referred to as a pump. So substances are moved again up the gradient of their concentration. So an example of a pump that would be relatable is a proton pump in our stomach. Okay, so as I mentioned before, our stomach environment is highly acidic. The cause of this acidity is the high concentration of hydrogen ions in the stomach. But we don't have high concentration of hydrogen ions throughout the body. So the proton pumps embedded in the membrane of our stomach lining, They are gathering those hydrogen ions that maybe pass by or are hanging out in the surroundings, and they move them inside of the stomach, okay, creating this artificial gradient, like it's a cell-made gradient of concentration, where inside of the cells there's more hydrogen ions than the outside of the cells. Another example of pumps or active transport is the sodium-potassium pump, which you probably encountered in grade school, but we will go over it again. So sodium-potassium pump moves ions against their concentration gradients. Why sodium-potassium pump is so awesome? It facilitates nerve signaling, it facilitates heart contractions, it facilitates kidney function. So virtually these sodium potassium cells are present in a lot of tissues within our bodies, and we need them to function properly. Okay, what sodium potassium pump does, in short, is it moves sodium atoms outside of the cells, or sodium ions outside of the cells, and moves potassium ions inside the cells. So the cells gather potassium and import them, and gather sodium and export it to the surrounding environment. So let's start with the first picture here. We have our membrane, we have the inside of the cell, or cytoplasm, and we have the outside of the cell. Note that the outside of the cell is rich in sodium ions and poor in potassium ions. So the sodium potassium pump, the protein involved in the sodium potassium pump, it has a certain configuration. So when the energy, when ATP approaches this protein, it donates the phosphate group. When the phosphate group attaches to the protein changes its configuration to let the sodium atoms, the sodium ions out of the cell. And it stays in this configuration for a while. It stays in this configuration until the two potassium molecules enter this transport protein, this facilitator protein, and once the two potassium ions docked inside the protein loses the phosphate group, and which changes the shape of the protein to the initial shape, and two potassium ions are imported into the cell. And after that is done, then we go to square one. When sodium ions are imported into the protein, energy is donated by the means of the phosphate group. The phosphate group changes the configuration of the protein, releasing the sodium, and potassium kind of floats in and is captured by the protein. Phosphate group is left, and potassium is imported into the cell. All right. Now, another two, I'm going to talk about the last two ways that cells export and import substances into them, and it's not going to be through the membrane anymore. So the two last ways are called endocytosis and exocytosis. So endocytosis, it requires energy, and what it is, it's basically a cell engulfing some external substrate into the inside of the cell. So the substance, once the substance approaches the membrane forms a circle around the substance, and it forms a vesicle. And this vesicle can then be digested by the cell. Remember, I think it was last lecture, Friday's lecture, the amoeba picture. Amoeba hugs are often deadly. So that's how amoebas eat. They eat by endocytosis. So they float around. Once they found food, they form this indentation in the membrane, form a vesicle. The vesicle eventually moves away from the membrane, and the other components, other organelles within amoeba digest this substance that amoeba engulfed. An interesting example inside our bodies of this endocytosis is white blood cells. White blood cells, they are part of the immune system. They circulate our body looking for things to defend us against, like viruses or bacteria. And once they found a virus or a bacteria, they do just that. They engulf it. So the indentation is formed inside of the membrane of the white blood cell. The virus or bacteria is then surrounded by this new membrane chunk, which is then separated from the membrane, forming a vesicle. And this vesicle then is digested by the white blood cells. And last way of transport is exocytosis. Exocytosis requires energy as well, and it is the opposite of the endocytosis. So whenever a cell has something to export, and remember, I gave an example of that at Friday's lecture when I discussed the protein formation. And remember that proteins eventually are expelled from the cell. And the example I gave was production of milk in the milk ducts of mammals. So the cells produce lipids and proteins, and I focused on proteins rather than lipids. And the proteins were produced in rough endoplasmic reticulum and then were transferred to Golgi apparatus. And from the Golgi apparatus, a vesicle was formed, and this protein was exported out of the cell. So this is an example of exocytosis. It's export of some substance from the inside of the cell to the outside of the cell by merging of the vesicle with the substance with the external membrane of the cell. All right, that is it. Before I move to photosynthesis, are there any questions about energy and transport? And feel free to unmute yourself to ask the question because it might take me a long time to go through chat at this point.
Does the sodium-potassium pump require energy?
Yes, so it is an example of active transport. And so all active transport or a transport down up the gradient of concentration requires energy. And you can see in the picture, you can see here that ATP is turned into ADP. So ATP is the energy currency and ADP is ATP without one phosphate group. I discussed it last time on Monday. And this phosphate group attaches to the protein. So yes, without ATP, there would be no phosphate group to attach to this protein. So this is where the energy goes in the sodium-potassium pump. Does this answer your question?
Yes, thank you.
Any other questions before I move on to photosynthesis?
Hi, Professor. A question from me. So for facilitated diffusion, it uses membrane proteins instead of energy, correct?
Yes. So energy can be even created in facilitated diffusion. So what the properties of facilitated diffusion is that the membrane protein has to be involved. The solutes move down the gradient of concentration. Oh, this is active transport, sorry. So in facilitated diffusion properties, again, the protein has to be involved in that is embedded within the membrane, and the solutes or substances move down their gradient of concentration from the area of high concentration to the area of low concentration. And in this particular example, the energy is created. The ADP adds the phosphate group because of the energy of movement of the particles from the area of high concentration to low concentration, forming ATP. So ATP is not consumed in this case, it is created in this case, but not in all cases, like it's just one particular example with the proton pump, or rather the proton movement. Did I answer your question?
Yeah, thank you, Professor.
Any other questions before I move on? moving on then. Photosynthesis. Photosynthesis is extremely important for our existence. Photosynthesis is the reason why atmosphere became oxygenated, or why oxygen was initially added to the atmosphere. All of the life on Earth depends on photosynthesis. Even though only plants and algae are able to perform photosynthesis, uh, the rest of life depends on the organic matter that is produced as a result of this photosynthesis. So, a little reminder that organisms that can capture energy from the sun are called autotrophs. Because auto means self, trophs mean feeding. So they're self-feeding organisms. They do not depend on other organic matter to survive. And photosynthetic organisms are producers, and the producers are the basis of every ecosystem on Earth. And as opposed to producers, we also have consumers who consume the producers and depend on the producers to survive, depend on the organic matter in the producers to survive. So photosynthesis converts energy. It's the process of converting the kinetic energy from the sun into potential energy potential energy stored in the bonds of the sugar molecule. So this graph I showed before, and I'm going to focus on highlight this portion of the graph that deals with photosynthesis. So photosynthesis is a series of chemical reactions that use light energy to assemble carbon dioxide from the air into molecule and water. Carbon dioxide and water from the air into molecules of glucose, the formula for which is C6H12O6. You don't have to remember the formula for glucose, but it's just useful to see once in a while. And other carbohydrates. So process that produces sugar is called photosynthesis. We touched on where the energy comes from during the last lecture while looking at this diagram. And the first step, basically, in energy acquisition from the surrounding environment is the photosynthesis. During the photosynthesis, some energy escapes as heat energy, because remember, no energy transfer is 100% efficient. And once the heat energy is lost, it cannot be recaptured. It's just the energy that, is useless to us. Well, it may keep us warm, but we cannot harvest it. As a result of photosynthesis, we have the oxygen and sugar. Well, products of photosynthesis are oxygen and sugars. And these are our products. And during last lecture, I was discussing what are the reactants. So can anybody identify the reactants of the photosynthesis reaction from this diagram? Okay, one
Are you?
version.
I
One
was
version
gonna.
is sunlight and heat. Another version, Dylan, go ahead.
I mean, I was literally just gonna recite the chemical formula.
Okay, so
CO2 plus H2O plus sunlight equals.
excellent. Yeah, I'm just
Yeah.
testing whether we know our terms here. Products are CO2, H2O, and H2O, carbon dioxide, and water in the presence of sunlight, and the products are oxygen and sugar. Great. Now, moving on. And sugar is a crucial food source, as I discussed yesterday on Monday. Animals, fungi, and other consumers eat the producers, and they depend on this sugar stored in the producers. Sugar is used in cellular respiration to make ATP for cells to use, and I will go over how that process is happening on Friday. So Friday's lecture will be dedicated for cellular respiration. Cellular respiration generates the energy for the consumers so that they can function, so that they can perform work. In our case, energy is needed again to breathe. Energy is needed for our peristaltics, which is the movement of the bowels that move food along. Energy is needed to run, to do any kind of movements, for the brain to work, and so on. So sunlight powers photosynthesis. Photosynthetic organisms need just a few simple ingredients or reactants to make their own molecules of sugar. And it's sunlight, carbon dioxide, and water. For instance, put a plant in the shade and see what happens after a week. And spoiler alert, it dies. Without sunlight, plant dies. If you deprive it of water, the second, another reactant here. If you don't water your plant, I'm looking at my ceiling here, windowsill here to see if I can show you, but I'm too embarrassed. I have a plant on my windowsill which I haven't watered in a while. It's a succulent, but you can literally lift it up and see the dry roots in there. If you don't water the plant, it will eventually die. If it's a succulent, it will take a long time to die. That's why I love succulents. But if it's not a succulent, it will die pretty fast. Within a week, I would say, of not watering, or 2 weeks. And the last reactant is the CO2. If the plants do not have access to carbon dioxide, they will also die because there will be no reactant to build the sugar molecules. There will be no carbon available to build this sugar molecule. CO2 is contained, or carbon dioxide is contained in the atmosphere, and you may have learned before that its concentration is increasing due to fossil fuel burning, due to burning of coal and gas and other fossil fuels. So as CO2 is increasing, it has been hypothesized that photosynthesis has been occurring at a higher rate. So the more CO2, the more food for plants, the higher is the rate of conversion of this CO2 into the sugar molecule. However, that hypothesis has been disputed lately because we don't see much increase in plant biomass in the areas that have not undergone human intervention. So there are some forests that have been intact for the past 50 years or 100 years. And typically, it's pretty rare to see the biomass in that forest increase in response to elevated CO2, elevated carbon dioxide. But I'll touch more on that. We'll discuss more about that towards the end of the semester when we discuss ecosystems. The the sunlight, is basically the photons that are defined as packets of light energy. Photons travel throughout the space in waves, and the portion of spectrum of light energy that reaches the Earth is in the ultraviolet spectrum, the visible spectrum, and we get some infrared radiation. So UV radiation, you may have heard about it. I don't know, you may have read about it a lot, and you may know that it's harmful. And that's why doctors recommend putting sunscreen, because ultraviolet radiation ruins our DNA in the skin cells, which leads to mutations and which leads to cancer. So put on your sunscreen. Infrared radiation from the sun is literally the energy of heat. We feel it as heat, but we don't see it, and we don't see ultraviolet radiation. What we see is this narrow band of energy from 400 nanometers to 700, well, a little less than 400 nanometers to 750 nanometers. And plants and other photosynthesizer capture the photons of visible light. So they capture the photons in this spectrum here. They don't capture ultraviolet radiation. They don't capture infrared radiation, but they capture the photons in the visible light. So the visible light is useful for photosynthesis. And it's 1:19 a.m., so 1 minute until the end of the class. So let me take a peek at the chat here.
Can I ask a question about the visible light?
Yes, go ahead.
So visible light is the only source, like light energy source, that can be used for photosynthesis? Or did you say
Yes.
ultra? Okay.
You're exactly right.
Okay.
So packets, meaning units. Sure, yeah. Units is another term for that. Packets makes it more visual, in my opinion. So you imagine like packets of light sent from the sun to here. But yeah, they're units of light. Okay, I'm gonna go through chat. So the reactants means the ingredients. Yes, Blaine, the reactants means the ingredients. The ingredients for the reaction and the products
I
for the
do.
reactions. Andrew, did you, or Dylan, did you say something?
With your nails. Here as you were.
So another question, does quiz one have the same questions in
Hello,
both
Pickies.
attempts? Yes,
I
it
don't like
does.
them right here. It makes
Okay, I'm gonna mute you. Okay, I don't see immediate questions immediately. So if you had a question, please unmute yourself and ask it.
So I think someone just asked this, and I'm sorry, but I'm gonna just ask again. So photons are the units only for visible light.
No, the photons also provide ultraviolet radiation and infrared radiation, which we don't see.
Okay, so all light is
Yeah, yeah.
measured in photons. Okay,
Yes,
thank you.
no problem. Okay, it's 10:21. I can answer any last minute question, and if there are no questions, this is the end of the class. I'll pick up photosynthesis and talk about respiration on Friday.
I have two questions. My first one is going back to the blood cells. I was just kind of like curious about the isotonic, hypotonic
Yeah.
with osmosis. Is if we have like too much water in like our blood cells, does that what causes blood clots or?
No. So blood clots are caused by not by this. Okay, this is interpretable. These pictures are interpretable when you inject something intravenously. So I gave an example of IV. Okay, sometimes when a person is severely dehydrated and delivered to the hospital, they're hooked up to an IV, and it is important to make sure that the solution in the IV, which you inject directly into your vessels, is isotonic. So it's typically a specific concentration of saline in that solution. Because if you inject water, this is what happens into the veins, like your blood cells become deformed. And if you inject the solution that is too poor in water, your erythrocytes or red blood cells will become deformed as well. But blood clots it's not how the blood clots are formed. Okay, we will go over it when we cover physiology and circulatory system.
Okay, thank you. And my second question was kind of just like a general question on the in-class participation assignment. Did we cover all the material for the photosynthesis in order to do that in-class participation assignment? I just wanted to double check with you.
You know what? I am not sure. I don't think so. So I'll go back to participation assignment, and if there will be a question. Yeah, so I will either delete the question, or if both questions were not covered today, I will change the due date to Friday.
Okay, thank you so much. I appreciate it.
All right, thanks for attending, guys, and I'll see you on Friday.
Thank you.
Thank you so much.